Hi, welcome back to Father-Offspring Interviews, episode 26. We've had a number of questions about aging and Alzheimer's and dementia from Natasha from Oregon, Andy from Pennsylvania, and then from some of your YouTube comments. So that's going to be the focus of today's episode. On the broader level, we're going to start with, what do you make of dementia and Alzheimer's in terms of where we are at with research?
Well, what do I make of it? I make of it that it's totally terrifying when you sit people down and ask them, if it's 2 a.m. and you're lying there and you can't get back to sleep and you're convinced you've got some horrible disease, what's the worst possible one? And Alzheimer's winds up at the top of the list. It gets about a third of people by the time they're in their 80s. It's the most common dementia. It's a total horror. What actually is Alzheimer's dementia? Everybody knows it's when you lose your memory, and there's a whole bunch of dementias. And what's with Alzheimer's? You don't lose your memory, you lose certain subtypes of memory. Short-term memory is okay. Remote memory, what was your native language when you were 5, that sort of thing. It's what's called long-term in between. That's the part that's damaged. You are perfectly fine memory of how to do stuff with your hands, procedural things. You get someone severely demented from Alzheimer's and they can't tell you what decade it is, but they can still take apart this gizmo, or they can still fix this or drive a car, which could be very dangerous. Things, what is termed implicit procedural memory, that maintains itself in there. The explicit declarative memory stuff, that's what gets damaged. So it's specific subtypes. As you know, back when you were just a twinkle in our eyes, Mommy was a clinical neuropsychologist working with Alzheimer's patients, and a lot of what she did was different types of memory tests to probe short-term versus long-term versus remote, explicit, implicit, trying to figure out the domains as to what was working, what wasn't. So what's the status of research on it? And here I think I'm going to be fairly cranky and opinionated and editorializing, in that I think the status of the disease, understanding it, is terrible. It is absolutely terrible. Alzheimer's is really 2 different diseases. First off, what's called early onset Alzheimer's, familial Alzheimer's. It runs in a family. It's due to a mutation. If one of your parents had it, you've got a 50% chance of getting it. Have the mutation, you're going to get the disease if you live long enough. And the characteristic of it is it gets you early in life. It gets you in your 50s, and it comes on and you degrade really fast, and you go from 60 to 0 in a couple of years. That's about 10% of the cases. In contrast, you're just garden variety Alzheimer's. 90% of the cases are what's called late onset. It's some memory problems in your 70s, and they become pretty seriously impairing. By your late 70s, by the 80s, you pretty much can't handle your finances anymore. You can't drive, that sort of picture. 90% of the cases, and as implied by the description, it's got a slower onset. It takes a longer time to incapacitate you, and it's not due to a single gene mutation. It's due to all sorts of stuff, and we don't really understand the all sorts of stuff very well yet. So you go to a conference on Alzheimer's disease, which I used to do a lot before I couldn't take it anymore because of how discouraging it was. And there'd always be a session one of the afternoons on therapeutics, what's new with clinical trials and all of that. And invariably, they would come back and the news would be, it didn't work, or the side effects are too severe. And these were always biotech companies. And then you could see all the scientists there scurrying out to go to the payphone to call up their stockbroker to sell their stock in that company or whatever. Really disappointing. None of the therapeutics would work. When I started going to these meetings way back when, okay, this one didn't work, but they're going to have this disease mopped up in no time. And many decades later, they still don't have it mopped up. Number of reasons. One is where my editorializing comes in. I think most of where people have been looking is the wrong place. Alzheimer's. pull out somebody's brain where you think they have Alzheimer's and you slice it up, and there's two things you look for afterward under a microscope. These things called amyloid plaques, which is made of this protein, the beta amyloid peptide, and it's an abnormal protein, where copies and copies and copies have gummed together and made this massive hairball crap gunk thing of a tombstone of an area where all the neurons are dead. So amyloid plaques. The other thing is you see these little tubules that run down the length of neurons for transporting stuff up and down, and they're all twisted up on each other. And those are called neurofibrillary tangles. And just to give you a sense of how much fun Alzheimer's research is, so the plaques made up of this rogue protein, the beta amyloid peptide, BAP, whereas these neurofibrillary tangles were made up of an abnormal version of a protein called the tau protein, TAU. And all of Alzheimer's research was divided into whether you thought the plaques were the problem or the tangles were the problem. Were you a BAP? Were you a Baptist? Or were you a Taoist? Can you believe how fun that was? And at conferences that would determine like who held the door open for who and who you could mate with. Were you a Baptist or a Taoist? And overwhelmingly, the field was dominated by the Baptists. Early on, back in, like, if you had asked me the cause of Alzheimer's disease when I was in kindergarten, I would have said it's a problem with a neurotransmitter called acetylcholine. And that's what dominated 70s, 80s or so. And it generated the first drug for Alzheimer's called Tacrin, which did squat. You give it to 50,000 people, and statistically you can show that cognition gets a little bit better. It was a Hail Mary, ooh, doc, can't you do something here? Let's try this drug. But by the 80s, the dominant model was it's the Baptist. It's the beta amyloid peptide, layers and layers of them forming these aggregate amyloid plaques. So everybody got hysterical about that and focused their research on that. And what was a first strategy? Make an antibody against the plaques that could come along and break apart the plaques or things like that. And this was totally exciting and tons of research on, you would have a single amyloid peptide, beta amyloid peptide, and once it started getting Velcro to more layers, its conformation would change. And what's the conformation that causes you the most trouble, and what other gunk gets caught up in these plaques? And the first attempts were to come up with make lab-based antibodies against the plaques. Whoa, inject that into people with Alzheimer's and that's going to break apart the plaques and everything's going to be great. There were problems. First problem was it caused a lot of brain swelling, like serious brain swelling in a subset of people. And the biggest problem was it didn't work. It didn't help at all. Ooh, I know why. We're not trying out this experimental thing until you have somebody who's got really severe Alzheimer's. Let's try it on earlier stage cases, and it simply didn't work. Massive disappointment, and people are still sticking with amyloid plaques. Then another type of baptism crept in. So this beta amyloid peptide, one single strand of a protein, it's a breakdown product, it's an abnormal, it's a diseased breakdown product of some other protein. So you generate this, and then a gazillion of them are going to layer together to form a plaque. Ooh, what's going on in between? You got one of these abnormal ones, and it binds to another abnormal one. You have what would be called a dimer. Then a third one, a trimer. These were not like tombstone plaques. They were still floating around, but you got this intermediate stage, and more people realized the intermediate stage is where the damage comes from. By the time it gets formed into one of these inert plaque things, that's after the fact. So everybody, all the Baptists, got very excited about these soluble oligomers. That's the jargon for it. It's just beginning to build up these things. They could begin to see these things could cause oxygen radical damage to neurons. They had all sorts of bad effects in synapses, things like that. So the hysteria at that point became, let's make antibodies against this intermediate form. And that's the state of the art right now. And basically, I think it's pretty dismal. The FDA has approved a couple of these antibodies in the last 2 years, and every sane, responsible person in the field wrote letters to the FDA saying, don't approve this stuff because it doesn't actually do anything. Once again, at 50,000 patients, and you could show statistically, 1.5 better points on a dementia test or whatever. Nonetheless, they were approved and they're all the rage, and they have a serious possibility of causing brain swelling. And even under the best of circumstances, they're slowing the decline down a little bit. So that's been this big problem with the field, and that they don't actually work all that well, which brings in the poor Taoists who everybody had been ostracizing at the meetings, the whole notion that these tangle things were the actual problem. This protein called tau would get abnormally folded in a way so that it would make the stuff like this, French braided abnormal thing. And what people first noticed was, Alzheimer's, you always see plaques and tangles. There's another type of dementia called frontal temporal dementia, similar in a lot of ways, also known as Pick's disease, where you don't get the plaques, you only get the tangles. Oh, I wonder if the plaques or the intermediate forms are bad news insofar as they give rise to the tangles, and any other means by which you generate the tangles, you're going to get into trouble. And people are finally beginning to focus more on that. There have been no breakthroughs so far, but I think that's where the best hope is. Yeah, still, there's no cure for it, and there's no way to substantially, reliably slow it down without a lot of side effects with very expensive medicines, all of that. Why is this? It's an insanely complicated disease where, among other things, what you're doing as a 20-year-old can significantly change the likelihood of you getting this disease half a century later. These are huge biological arcs of stuff. Good luck figuring out how that works. And a gazillion really good scientists have been working for a long time trying to figure it out. Last issue with why there's been so little progress. And this is one where sort of showing science's occasional dirty laundry, but there's been two very notable scandals in the field where findings that were breakthroughs, that this might be it finally, turned out to have large elements of fraud in them and had to be retracted. The first one was a suggestion that these little beta amyloid peptide fibrils could turn on what are called death genes and neurons. And whoa, that was going to explain everything. And I taught this paper for years. It came out of a group at Genentech, and it turned out a mixture of data that were highly unreliable, let's say that kindly, and administrative lack of enthusiasm to let the world know that this wasn't for real. So that got retracted. The other was a group, University of Minnesota, that looked at the first intermediate form that they had stabilized and said, this is the one that kills neurons, and we can purify it, and this is going to... And it turned out they didn't quite... And again, some question of ethics, and that was retracted. So that on top of it, whoa, is this a higher rate of fraudulence than in other fields of science, let alone CEOs embezzling funds? Answer is, I don't know, but two factors here. One is if you've just made up data about like the mating rituals of spotted geckos or something, it's probably going to be 80 years before somebody else studies the subject to find out if it's replicable or not. This stuff, a new finding, everybody is immediately on it. And the other is, this is the most frantically manic group of researchers out there. Manic because this damn disease. Manic also because whoever gets this is going to make Jonas Salk look like nothing in terms of solving this tremendous, tremendous pressure to get the breakthrough, even though there's not going to be the breakthrough. So overall, a pretty grim picture. At 30, when I would go to these meetings, I would say, well, that's okay. They have no idea how to cure it yet. I got plenty of time. they're still doing that, and I don't got plenty of time left now. It's a pretty grim picture.
So now someone also said, I've been a musician all my life. Did practicing and playing music in my younger years help guard against Alzheimer's and dementia that my mother suffered from in her senior years, or is my DNA going to eventually catch up to me?
Yay, musicianship can definitely help you. Here's about the only good news in here. Okay, back to the previous one. 10% of cases are the early onset genetic ones where you get the mutation, you're going to get it. It's a sledgehammer, or the non-genetic, non-familial, late onset one that gradually emerges. So 90% of the cases are the gradual late onset. 90% of the research has been about the early onset because it's cleaner. You could figure out the whole damn disease, identify the mutation in this family. It is so flashy, and the late onset stuff is just such a mess. It's not caused by a gene mutation, and it's a total quagmire. Nothing you're doing as a 20-year-old is going to affect, if you've got the mutation version, stuff 20 every year thereafter. And it's been like this nightmare of trying to make sense of the late onset. One exciting hint, one way in which genes crept in, was a gene where there's not a mutation. Back to early onset, have the mutation, you're going to get the disease. This was a gene that instead came in 3 different flavors, and which flavor you had determined the likelihood of you getting Alzheimer's. Not certainty, not causal, just modulating the risk. And this was a gene for something called apolipoprotein E. When this was first discovered, every neuroscientist on Earth immediately went to like some old textbook to find out what in God's name apolipoprotein E did. It's related to high-density lipoproteins, and no one knew where that fit in, but there were 3 different versions of ApoE, E2, E3, E4. I don't know if E1 was discontinued or something, but it turned out if you have E2, you have maybe a 15% chance of getting Alzheimer's. E3, about a 50% chance. E4, like an 85 to 90% chance. Like, not absolute, but hugely modulatory. So everyone got excited about that, and tons of work went into finding out. So what's the difference between E2, 3, and 4? And years later now, an astonishing amount of ignorance is still there, what that has to do with making plaques or tau getting tangled up. It probably has much more generic things to do with brain health during aging, but that's been kind of a disappointment in many ways. And that was 3.5 hours where everyone read that first paper and said, that's it. Disease is going to be solved in no time. So not a whole lot gotten from that, but you can go and get yourself genotyped and find out which version of ApoE you have and plan accordingly, and keep in mind you were looking at probabilities, not certainty, all that sort of thing. Okay, nonetheless, ApoE, a genetic modifier, was the flashiest, most exciting thing in making sense of the late onset one. And then you'd realize for years you'd go to these Alzheimer's meetings, and the very last session on the last day would be something like psychosocial interventions in Alzheimer's disease. And everybody would like roll their eyes or check their emails, or this was their chance to go out and get the Danish or the donuts, or see if they can get an earlier flight home, because God, 3 hours of a bunch of psychologists and a bunch of social workers and like a professional group hug facilitator are going to get up and tell you about Alzheimer's disease. And what people have figured out in these years is when you look at the late onset, the one that's going to do in 90% of people who get the disease, about half of the risk is from lifestyle risk factors. About half of the risk is modifiable, which is like exactly the opposite of your Mr. Machine scientist who are saying, show me the mutation, the lifestyle risk factors. And anyone who is paying attention is realizing this is the most exciting thing that Alzheimer's a long time. Doing things in how you live your life over the course of decades and decades can very significantly can explain half the variance in the risk of this disease. Okay, so what sort of factors come in? First off, above all else, never be a professional football player or a boxer or have a serious concussive head trauma, a traumatic brain injury. Even as a 20-year-old, any sort of concussive injury greatly increases the risk of a dementia later on. And it turns out traumatic vibrational impact to the brain generates a lot of that tau stuff. And okay, so don't do that. Don't do that. And if at all possible, avoid that. What else? You should try not to be clinically depressed, because decades of untreated depression turns out to be one of the significant risk factors. You should not spend decades getting 2 or 3 hours less sleep than you actually need. It turns out when you're asleep, this wonderful sort of system discovered University of Rochester a few years ago, where basically at night, your brain flushes out all sorts of gunk when you're asleep. And some of the gunk are amyloid fragments and tau fragments and things like that, and less sleep puts you more at risk for that. So that's not good. Next, if you want to be protected, have really healthy blood vessels, especially in your brain. Wait a second, what's that got to do with Alzheimer's? Alzheimer's is these microscopic plaque things and these tangle things. And what is your blood vessels? Because it turns out that's another modulatory factor. Have X amount of messed up dying neurons because of plaques and or tangles, and whether or not you are going to have a clinical profile of Alzheimer's is much more likely if you got lousy vasculature, if you've got if you got clogged vessels. So that's part of it. So immediately we're getting into diet and smoking and alcohol and stuff like that. Alcohol abuse all on its own forms its own kind of dementia, but it almost certainly increases the risk of Alzheimer's. So that's in there. What else? Exercise. Exercise is like one of the best things you can do from the time you're a toddler until later on in terms of, well, it makes for a healthier cardiovascular system. It also does all sorts of good things directly to neurons, making them a little bit more resistant to some of the stuff that goes wrong in Alzheimer's. So that's great news. Other great news, make sure you're well educated when you're young. Oh, bummer. Let's go back and fix that one up. Degree of education early in life is a predictor. Is that because education slows down your rate of brain aging? Or is it merely you start higher up and you're decaying at the same rate? You got more marbles in the bank and you can afford to lose more of them. And it's probably a mixture of both. But what is clear is intellectual stimulation your entire life makes neurons grow new connections, makes synapses that are already there more excitable, probably makes you make new neurons. And these are the parts of the brain that are carpet bombed in Alzheimer's, the hippocampus and the cortex. So that is hugely exciting. What's implicit in being intellectually stimulated is playing a musical instrument, learning a new language. Musical instrument, unless you're like a double reed player, in which case you're probably doing back pressure to your brain from those weirdo oboe things. But any other music thing, music, a new language, all these things, including in people at age 90 or so, put 90-year-olds in a more enriched environment, and neurons there will grow new connections. And years ago, my lab was working with some genetic models of Alzheimer's disease where you introduced one of those mutations into mice, and they developed plaques and tangles and cognition fell apart, and put them in a more enriched, stimulating environment, and you delayed the process. Okay, what ultimately is probably the best thing you could do for mental health and brain stimulation, all of that? Social support, social affiliation, and loneliness intertwined with depression, intertwined with bad health habits that are a function of that, etc., etc. That's one of the biggest ones out there. Oh my God, loneliness. And we're talking about like the molecular biology of amyloid and Yeah, it's playing out on that level. The final thing that comes in there is one that had me dip into the Alzheimer's field in the 90s or so before I burned out on it, which is the possibility that stress over the lifetime can increase the risk of Alzheimer's. And we saw things back when, like stress and elevated stress hormone levels would make for more tau proteins that are wrapped up, things like that. Large epidemiological studies now showing stress, which is kind of a surrogate for depression, which is kind of a surrogate for poor health care access thing. Stress appears to be one of those factors also. So all the ways in which things can go wrong in you if you don't do it right. But what's amazing news is like half of your risk for Alzheimer's disease has to do with modifiable things in your lifestyle. So to get back to the original question, yeah, playing your musical instrument all these years is great, is exact. Keep doing it. Do it with friends. Do it in a challenging way. It's great. Perfect.
Then finally, how does dementia manifest in other primates and species?
Well, classic sort of question in this realm. Are we alone? Are we the only ones who get Alzheimer's? Alzheimer's, under the microscope, amyloid plaques, neurofibrillary tangles, amyloid plaques that are concentrated in the hippocampus and cortex, lots of dead neurons, and, as described at the beginning here, dementia. A massive cognitive decline, completely incapacitating, where ultimately the person can't tell you the name of their spouse of 60 years, what decade it is, etc. So do you see something like that in non-human primates? And basically not. You see some amyloid plaques develop in the brain. In primate aging, these other species, there's not as many, and they're not focalized into hippocampus and cortex. You see the same weirdo versions of the tau protein as you see in us, but in virtually every study, you don't see the tangles. Something like that doesn't happen. And most of all, you don't see massive cognitive declines in non-human primates, and you give them the equivalents of some of the human tests for memory and different domains of memory and such. So why is it? Why don't they get it? One possibility is, you look at a primate that lives for 30 years, some monkey. Ooh, there's simply not enough time. The odds are that's not what's going on. There seems to be different conformations of the tau protein in non-human primates than in us, but mostly the answer there is no, you don't get Alzheimer's. Do you get typical brain aging, though, that we recognize as typical and normal in us and non-human primates? Absolutely. As they get older, cognition gets worse. Long-term memory problems emerge, executive function, all this stuff that I mentioned earlier, that absolutely emerges. So you don't get Alzheimer's, but you got brain aging, and that's what goes on. And some years ago, this gerontologist named Caleb Finch at USC, he and I came up with a somewhat crackpot theory about the evolution of Alzheimer's disease built around brain aging in normal natural populations of primates, including us. So if you were going to talk about brain aging in wild animals, including primates, you're talking about females. Old age is not common in males, so we're just focusing on females here. And we noticed something interesting in the literature. Primates, female primates, only a subset of them get menopause. Something is clear cut as the whole system shutting off. But all female primates, as they age, get at least a decline in fertility, a big decline during a period with that. And amid all sorts of primate brains getting plaques and abnormal thises and that's and inflammation and things of that sort, what we noticed was, interestingly, you didn't see a whole lot of that until after these females had gone through their period of reproductive decline. And that's when you saw the damage emerge. Okay, so what's that about? We made up this totally satisfying just so story as follows. If you're a giraffe and you were just born and you plop 8 feet out of mom and you land on your head. You then get up and shake yourself off a little bit, and then you're off and running and functioning. Most species, animals are fairly together soon after birth. Primates, not just us, not just apes, but all the primates take forever to mature because they gotta grow these fancy brains and social complexity and all of that. And one of the things going hand in hand with that is this work by someone named Sarah Hrdy, pioneer in this UC Davis, showing competent primate mothering is not instinctual. You got to learn how to do it from your own mother, from your big sister. If you have a big sister who you could observe and learn from, your first child is more likely to survive, things like that. So it takes maternal smarts, knowing which valleys got trees that are fruiting this time of year, and what do you do when there was a drought 10 years ago, and you need maternal competence. In other words, things are not so good if you start in female primates getting a significant decline in cognition while still during a reproductively active period. So the just so story is, it would be great if we could invent some sort of signal for the brain saying, hold off the pathology, hold off, stop the plaques, don't have things go wrong until we wean our last kid, until we've gotten through that period of having to be a competent, intact mother. And so how could you do that? And it turns out the thing that does that is estrogen. Estrogen has all sorts of great effects in the brain. Estrogen works as a powerful antioxidant. It causes neurons to express growth factors, things of that sort. This puts it into a minefield of what should be done in terms of postmenopausal estrogen replacement, if it should be done at all. And that is incredibly controversial because it's a really messy question. Estrogen is not a single hormone. It's a family of them. Estrone, estriol. So what combination of estrogens? Is it synthetic or is it natural? Do you have it with progesterone or not? At what doses? And probably most importantly, do you start someone on it right at the start of menopause? Or do you wait until they've gone through menopause and then start it? But I think what the non-human primate studies show is you get the closest thing to the natural hormone profile and start it when the decline is just starting, and it's really protective against the cognitive declines and brain aging. Okay, so hooray, that explains everything. That's wonderful. How can you still be a competent primate mother when you're getting along in years and you got your last child? Estrogen has been holding off the pathology, and that's all sorts of cool stuff with that. But then you get to us, and there's a problem. It's not that, you go through menopause in your early 50s and you manage to get your last kid off to pre-K or something, and then suddenly you got memory problems. We don't get these slow dementias until 70s, 80s or so. There's something different in humans because estrogen has long since gone down. What's the deal? How do humans get an extra generation worth of cognitive intactness? Just so story. So why might that be a useful thing? Yeah, you want to have mom being competent, but why do you want to have competent grandmothers? And this brings up the work of an anthropologist named Kristen Hawkes, who sort of pioneered this, showing that humans have grandmother effects. You look at traditional human cultures and having a maternal grandmother around, if it's a matrilocal society, having a maternal grandmother around greatly increases the likelihood of grandkids surviving. How come? She helps out. She pays for your piano lessons. She helps with foraging. She helps with all that sort. Having a maternal grandmother around is very, very helpful. And in fact, what studies have shown is among traditional hunter-gatherers, grandmothers provide as many calories to the family as the father, the great hunter, provides their grandmother for. So grandmothers are really important to have around and to be cognitively intact. And thus the key question is, do you see the grandmother effect in other primates? And no, you don't. It's a human invention. Okay, so we've come up with a social system where having cognitively intact 75-year-old grandmothers really helps copies of genes get passed on in terms of child survival. So, whoa, let's invent a way of like getting an extra 20, 30 years worth of cognitive intactness out of brain aging. How are you going to do that with our Just So story? And it turns out there's an explanation. Back to that apolipoprotein E, E2, E3, E4. Have E4, 85-90% chance of getting late onset Alzheimer's. E2, you've got like 15% chance, E3 in between. And you say, oh, well, I sure would like to have E2 or E3. What's the deal with that? And you look at the evolution of this gene, and we humans, we're the ones who invented ApoE2 and E3. E4 is found in every other primate. These more protective variants emerged in humans in the last 300,000 years or so. What is that? That's something we've been evolving to get more deal with the social complexity that can arise from having cognitively intact grandmothers around. That's totally great. Ooh, are we in the process of seeing everyone's descendants eventually will be E2? Probably not. E4, bad news, brain aging. Turning out E4 early in life has some antimicrobial actions. It increases progesterone and fertility. So you got a trade-off. How much protection do you want early in life? And along comes a bill later. Or how much do you want to save the protection for later in life? A balancing of it, and fascinating stuff looking at where in the world you see the highest levels of each subtype. Okay, so non-human primates don't get Alzheimer's disease. They get brain aging just like us, except we do it completely differently because of this ApoE evolution business. We can have 75-year-olds who are still cognitively perfectly fine, and they're great to have around for the grandkids.
And that is it for episode 26. Keep submitting your questions at the form found in the Instagram story highlight and bio or the YouTube video description. I'm Offspring Sapolsky, and thanks for your continued support of Science and the Beard.